So that could be an issue, but it's probably not as big an issue as if you use up all the ATP. Any other questions? Yeah.
Yeah, I keep getting the idea that it's a all or nothing. If it's going on and one pathway is basically constantly running and then it shuts off and then another one kicks in.
Yeah.
But is it like that, or is it constant like shifting up or down to maintain that kind of level of homeostasis?
It's probably not. It may not be completely turned off, and maybe it's effectively turned off to if you were to compare it to the other one. So that it all depends on the levels of the allosteric inhibitors and how effective a job they're able to do. What is true is that when you turn on this one, right? If you're in a low energy state and you want to turn on this PFK, you still don't turn it on to the extent of full activity, right? There's still enough balance in the allosteric effectors to keep it at a low activity, right? So it's kind of like you can think of it as fully off or partially on, but the extent to which it's partially on depends on the details of which reaction it is and whether, for example, you're implicating the ATP stores, right? So you could look at it like that.
This might be outside of your wheelhouse, but I'm wondering if you know physiologically how long it takes for an organism to switch over from glycolysis to gluconeogenesis.
Not long. Certainly on the time scale of minutes, if not faster. Probably faster. Because everything is about, well, it depends on the kind of regulation. No. The kind of regulation we're talking about does not involve, for the most part, anything to do with genetic expression.
So we have all of the proteins.
In other words, yeah, like if we may have a kinase and a phosphatase, which would involve a covalent modification and the removal of that covalent modification. But if the kinases and phosphatases that do that are also available, then they're there, right? The small molecule allosteric effectors are there, typically.
So under actual conditions, we might have to go make some of these components, recruit more things, express genes, but with our model, we're saying it's all ready to go.
Yeah, yeah, it's safe to say it's all ready to go, and then that's a relatively fast reaction. And the slow reaction is when you need to actually synthesize something. Like a slow thing clearly would be the cell is going from a progenitor stem cell into a differentiated nerve cell, and you need a whole transcriptional program, and that's clearly slow. And there are other events that, for example, external hormones that may trigger not an immediate event in the cytoplasm, but a signal that gets into the nucleus and turns a gene on. That would be relatively slower as well. Yeah.
So with the PFK and ATP, so ATP is the effector, is that right?
ATP is both a substrate and an effector. It's coming up on a slide soon. Yeah. Yeah. Yeah, it's a little tricky in that case because the ATP is a substrate. So. Okay. So the other sort of general point is to look at this flux control analysis, and so I kept that in your reading. I think I excluded on the syllabus that complicated box where there's a mathematical analysis. But increasingly work in enzymology is turning towards pathway level analyses and looking at how the regulation works in the context of the whole cell rather than thinking about individual enzymes. And so this kind of analysis, this sort of metabolic control analysis, is an approach to do that. And so this is in your book because the authors would like you to get a sense of what the direction of the field is like. And clearly the direction of the field of enzymology is moving away from detailed mechanistic study of individual enzymes to more global pictures of what makes a cancer cell different from an untransformed cell, really. What are all the metabolomics that are different? If this pathway is turned on, which effectors can work on it, and how do background genes that you get in cancer affect that because they change the proteome, they change the mRNA levels, right? So the experiments end up looking at things like what proteins are present of the thousands that could be, what are the messenger RNA levels, and how fast are the pathways working So this sort of analysis is really very much of a model system in the sense of, you know, glycolysis in E. coli. Right? So this is not something of particular medical interest, but because we understand E. coli really well, we understand glycolysis really well, if we do this experiment in that kind of system, we can at least see what the, you know, what the capacity of these experiments is to give us information, if you will, right? So typically, when you do, the most important thing here is the flux control coefficient, right? So this is glycolytic flux and how much enzyme is added for 3 different enzymes. And this is the kind of analysis that depends on everything that's going on in the pathway. It depends on all the substrates, all the effectors, all the enzymes, and the levels of these things. So the thing is chugging along, and you've got an E. coli, it's going along in an extract, okay? So it's not really in the cell, but it's in an extract. But everything is there and everything is working, right? And so it's working in the direction of glycolysis. And so the idea is to ask, what happens when you add specific purified enzymes into the extract in excess, right? So if you consider glycolysis and what's regulated and what's not, and you've got an extract and the glycolysis is running, and you can see it's running because there might be a little ATP coming out, or there's some other measure of how you know it's running. And then you add, you know, some amount of extra phosphohexosaisomerase, which is step 2. Right? So we know that's not a regulated step. It can go either direction. So we add a whole bunch of that, and think about what would happen there if we were in this kind of situation, right, where phosphohexosaisomerase is like aldolase. So let's say we add 10 times as much aldolase to this. What will the effect be? Zippo, right? Because 10 times as much, it's not a driver, right? If you have 10 times as much of a driver, right, then the flux, the actual throughput, is what we're talking about here, right? Not the relative amounts, right? If you don't change the effect, the allosteric effectors, and the substrate and the cues, right? You don't change the balance of this. But if you have a lot more of this enzyme, it can still process a lot more of the substrate, right? So that's kind of the idea. It turns out, and it's a little counterintuitive, everybody thought, well, the key regulation is on PFK in this pathway, because that's like the committed step, not necessarily the thermodynamically irreversible step of all steps, but the step after which the cell has nothing else to do with the metabolites except shoot them through glycolysis. But it turns out that that's not the key thing. You add more phosphokinase, you get a little bit more forward flux of glycolysis. More pyruvate comes out the end. But not nearly so much as if you add extra hexokinase. So that was a little counterintuitive and interesting, that the regulation seems to depend more on hexokinase than it does on PFK. So, you know, we can think about that in the big scheme, and that would be what this kind of experiment, you know, allows you to do. There's other parameters. There's this elasticity coefficient, epsilon, which measures how responsive an enzyme is to its different effectors. So that kind of experiment, you'd be doing the same thing, but you'd be adding, you know, for example, a lot of, we'll see citrate is a signal of high energy state, and it feeds back to inhibit certain enzymes and not otherwise. So that would be a way of looking at, well, what's going on with that enzyme? How important is that particular allosteric effector? Right? How important is AMP, for example, in regulating this overall pathway? And then response coefficient, impact of an outside factor, so some other kinds of control, like a hormone or something like that. Not really that different from the elasticity coefficient, conceptually. There's an equation in the book to relate these. It goes a little bit beyond what I think we need to think about here, but it's just to sort of give a sense of what sorts of experiments the field is getting into. Yeah.
I'm sorry if I don't understand, but is this level of analysis done in human cells and looking at specific pathways?
Yeah, it's possible to extend the analysis into an in vivo situation, and it's certainly a lot more interesting to do that from the standpoint of, you know, a pathway that's involved in for example, tumorigenesis. But it does turn out that these very basic energy pathways are completely messed up in